I've worked with a lot of large drug companies, medical device companies, and sure, they invest a lot in the development of those products. But if you look at the origin of the ideas behind them, often they've started as NIH support. So when you see these massive cuts, I worry about the long-term impact that has on both economic development and the health of our population.
Hello, I'm Bruce Berman, the host of Understanding IP Matters, the critically acclaimed podcast series that provides leading innovators and experts a space to share their IP story. Learn firsthand about the strengths and weaknesses of copyrights, patents, trademarks, and trade secrets as they adapt to an AI-driven world. This is our 4th season. Welcome to another episode of Understanding IP Matters. I'm your host, Bruce Berman. We have been fortunate to have had a range of guests on Understanding IP Matters: inventors, strategists, entrepreneurs. Today we have someone who is that and more. He is a medical device developer who is addressing diseases like cancer and epilepsy by circumventing the blood system and going directly to the site of the disease. We are today with Michael Cima, a celebrated researcher, inventor, and entrepreneur responsible for creating advanced medical devices. He is also a devoted educator, mentoring the next generation of creators about the critical role IP rights play in innovation. The Cima lab at MIT focuses on diagnostics and treatments for cancer, metabolic diseases, and trauma. Many of the devices designed and engineered by the lab are injectable and minimally invasive. Dr. Cima is lead or co-recipient on 58 patents. He is Associate Dean of Innovation for MIT's School of Engineering and Faculty Director of the Lemelson MIT program, which seeks to inspire young people about inventing. A serial entrepreneur, Dr. Cima has founded several companies, including 2 with Robert Langer, the world's most cited engineer, credited with 1,600 papers and 1,500 patents. Their collaborative work includes Microchips, a developer of microelectronic-based drug delivery and diagnostic systems. They are also co-founders of T2 Biosystems. Good morning, Dr. Cima. May I call you Michael?
Yes, please.
Okay. Where are you speaking from today?
I'm here at MIT in Cambridge, Massachusetts.
So let's get right into it. Traditionally, university researchers were not focused on licensing or starting new companies, let alone building them. What has changed?
The one exception to that may be MIT, right? Because there's a long history of what we call translation here.
Tech transfer.
Yeah, tech transfer. It's sort of built into the ethos of the place. I think in general in academics, since the Bayh-Dole Act, universities have seen their role change. They've been incentivized to be players in what I would call economic development. They have to take their role of economic development seriously. And part of that is translating technology.
You started your career as a chemical engineer. How did you become a medical device entrepreneur?
It's interesting. I've never taken a biology class. I like to tell students that. But the world around MIT changed. I mean, next month will be my 39th year at MIT. And if you think back to the late 80s, I started in 86, obviously. That was the beginning of the biotech revolution around here. The following 15 years, the environment around MIT changed. So much so that MIT administration realized that MIT had to play an even bigger role in the biotech industry. For example, our dean of engineering at the time, a guy named Bob Brown, who subsequently became our provost, he decided we should have a biology requirement for undergraduates. Then he said, interestingly enough, in a year or two, we're going to have a situation where our undergraduates know more about biology than our faculty. So he created a course for faculty. And it was a who's who list of instructors. There were Nobel Prize winners and stuff like that. It was kind of a laid back course, but it actually had a lab. And I took it, and it gave me the sort of confidence to read more about the area. So that was an important factor. I did have a little bit of an education. And now, when I started at MIT, I was involved more in electronics. And specifically, I was involved in powder processing for things like capacitors and things like that. So my translation journey really started not in medical products, but in what today we call 3D printing.
Interesting.
And at the time, we called it solid freeform fabrication. And in the late 80s, there was not much around. I mean, there were like four or five other groups in the world, and I knew everybody because, and it was that was at that time the basic. There's probably four or five different solid freeform fabrication technologies. They all started in a roughly the same time, and it was primarily because that was the era that solid modeling became part of the prototyping process, the design process. And solid models, as opposed to drafting, made it much easier to convert that design into instructions for a machine. And that's really was the catalyst for all of.
I've worked with many patent attorneys and maybe hundreds, and often they're chemical engineers. And I find them the smartest folks that I've met in business and the most humble as well, which is ironic. Coming out of the arts, for me, it's always what you've read, where you've been, who you know. But I don't think chemical engineers don't need that.
Yeah, they don't need that. Right. Yeah. Yeah. My background was chemical engineering. I mean, PhDs in chemical engineering. And then I came to MIT to teach material science. And so the first material science class I took was one I taught. And I've been doing that for almost 40 years now. Long story short is, we were very successful at translating 3D printing. I mean, we had 12 licensees by the peak, and we had millions of dollars worth of research funding and lots of students. But at that point, we realized there wasn't much we could do at a university. I mean, we were competing with our own licensees who could hire professional engineers, and my co-inventor Ellie Sachs and I had students. So we kind of decided to phase out of that. And I went on to start prototyping not shapes, but formulations. And we ended up with Bob. We started a company called Bob Langer. We started a company called Transform Pharmaceuticals. And what we were doing was building on a change in the regulatory environment, which required companies to prove that their new drug was in a stable polymorphic form. And most drug companies really didn't understand what polymorphs were. And they had to show that they diligently evaluated all the polymorphs. No one knew at the time how to create all the polymorphs. And so that's what we did. I turned from making shapes to making formulations, and we were very successful at that. By the time that company was acquired, all the major pharmaceutical companies were our clients. When it was acquired, it ended up being all our clients had to develop all that themselves because they couldn't get access to it anymore. And so now I can confidently say every new drug developed using a system like what we developed.
About what year was that?
So it was acquired in 2005, 2004, 2005. During that time, I was working on the most difficult compounds at every major pharmaceutical company. The ones that they couldn't crystallize, or they had little bioavailability. It was a great learning experience. And then the company that acquired Transform Pharmaceuticals, part of the deal was I had to sign up as a consultant for them for 5 years. And I got to work on all kinds of problems all over the world. So it was, despite never having taken a biology class, I got a great education on real problems.
You're associated, Michael, with 58 issued patents, I believe is the last number I have for you. At the same time, I believe you're also co-author or author on 250 papers,
Over 300 now.
over 300 now. How do you resolve publishing with patenting, which discourages prior disclosure?
You said, I mean, particularly in the medical products area, you are, let me put it this way, you're forced, intellectual property is incredibly important because you're forced, if you're doing experiments on people, you have to disclose what you're doing, right? So the thing that you do right up front is file patents.
A provisional, at least, or if not a full patent.
Yeah, I mean, you have to. I mean, it's part of the landscape, and you have to do it early. Part of the downside is you're doing it, you could argue you're doing it too early.
Is that done through the university, or do you do that on your own as a business?
Both. So if it's done based on work that I do here, sponsored research at MIT, MIT owns the intellectual property. And so I work with, we call it our technology licensing office, TLO. Their process is they have teams of lawyers that they work with to do the filing. And I've worked with Sutherland for decades now on all the drug delivery type patents that I've worked on, and it's Kevin King there has become a good friend of mine. So we worked together so long, and so we kind of think alike.
He's educated you, and you've educated him, I'm sure.
Oh, absolutely. But because he knows the art, it's very easy for him to write, even improve the specifications to make sure that we are disclosing things appropriately. And I mean, I've gotten pretty good at it. The other thing that I've done on the intellectual property side, and I know Bob does the same thing, is I serve as experts in IP litigation. And I don't do much of it. It's like maybe one or two a year, but that's been very important for me as an inventor. Why that is because you learn a lot about how to defend a patent by going through litigation. So when I'm working with Kevin, I'm thinking 10 years down the line when this product is incredibly expensive. And of course, there's going to be litigation about it. I'm thinking about that, what that defense of the patent is going to look like when I'm writing my own patents.
You think like business, and you think also as a litigator.
Exactly. The litigation attorneys I've worked with have been actually instrumental in my growth as an inventor because they really point to the weaknesses and things like that you don't think about until you've had that experience.
That makes you a better inventor. Recent government cuts, Michael, to university funding will slow research progress. How concerned are you about the loss of funds, and how do you think researchers plan to deal with the shortfall?
This is an incredibly important issue. Depending on the actual magnitudes of these cuts. I mean, it's not just on research support. There's kind of an attack on academia. It's one of the institutions that, people want to question whether these institutions have served them. And they don't really necessarily touch the kind of economic development things that I was referring to earlier. In the medical products area, it's kind of hard to point to just about any new medical product that wasn't touched by NIH support. And it leads in its early conception, right? I mean, I've worked with a lot of large drug companies, medical device companies, and sure, they invest a lot in the development of those products. But if you look at the origin of the ideas behind them, often they've started as NIH support. So when you see these massive cuts, I worry about the long-term impact that has on both economic development and the health of our population.
How do you think the shortfalls will be made up through endowments or through private equity funding or what?
Can we make up the monies that they're talking about are massive. I don't think it's can be done overnight, but I do think we are moving. I don't like we'll have to do that over some period of time, 5, 10 years. We're going to have to replace that. And so there's going to be philanthropic sources. We're going to have different kind of partnerships with large companies. I'm not sure that that's going to give you the best bang for your buck because those will all come with strings. And if the federal government goes away, we will find a way to do it. But I do worry that the MIT that we've grown to, and I speak, like the rest of academia, but especially MIT, it's going to be hard to be the same place that it was when you're talking about like 50% cut. In the worst case, I mean, because it wasn't just the NIH funding, it's the indirect rate is being slashed. That does pay for research. It's hard to believe. I mean, in fact, MIT subsidizes research even at the high indirect rate. You're seeing proposed tax on endowment return. If you added all that up at MIT, it could cost as much as $800 million a year. How do you plan for something like that? Now, I just saw over the weekend that, at least in the Senate version of their budget, that MIT may not get the 21% tax on. It's going from 1.5% to 8%, or I don't know where we're going to end up in this because probably nobody does. But these are huge numbers. This is not like something you can plan for and just overnight replace.
It's going to be a challenge for years. You were co-founder of T2 Biosystems with Bob Langer. Langer is one of the most prolific inventors in history. His work, as you know, in biotechnology, drug delivery, and other areas has resulted in more than 1,400 patents. I think it's 1,500 now. And he is the most cited engineer in history, I believe, and one of the 10 most cited individuals in any field, having authored over 1,600 papers. What's it like working with Bob Langer?
Well, I've known Bob since I started at MIT, so almost 40 years. We've had a fantastic working relationship. I'm on the 6th floor of the Koch building at MIT, and so is he. We have a great working relationship. We're personal friends, and we've co-founded several companies.
What part do you do, Michael, and what part does he do? How do you divide the workload in that context?
It just depends on the project. I'm on the most recently, the one we exited was Taurus Biomedical. That was a couple of years ago. It was acquired by Johnson & Johnson. We were co-founders of that. It was basically, I brought the technology in that one. It was almost 100% from my lab. But Bob and I have had a strategy on how to get it funded. And that really worked well. He was exceptional at getting that to happen. So we both served on the board there. It was acquired by Johnson & Johnson a few years ago, and the product that we developed, they continued to invest in and filed a new drug application this past January. And I think this summer there's a chance that it'll get approved. That's for bladder cancer.
Oh, that's great.
The data looks spectacular. It's now that was a 15-year project. These projects are really long, but we went from what I would call basic research, preclinical research in the lab here. Your listeners will probably enjoy that story because I was just telling a group of students about it the other day. The project was to deliver drugs to the bladder, and it was inspired by studying what urologists do. Bob actually introduced me to a urologist, And it turns out urologists have a practice of making a drug solution off label, catheterizing a patient and filling their bladder drug solution. And they tell the patient, hold it as long as you can. That's their exposure to the drug. And for a number of indications, everywhere from inflammatory bladder conditions to bladder cancer. And I thought this is kind of crazy because, especially for cytotoxic agents, that's probably not a good way. I mean, this is how you might breed resistance to the drug. We started thinking about how will we do that. My group started thinking about how to do it. And I had a whiteboard in the office. Actually, I had a conference room in the old building I was in. And we would start putting down ideas. And we looked at every procedure a urologist does. What tools did they use? What things are in the bladder? Catheters and stents and things like that. The hardest one was, what if things people put in the bladder that were not successful? In a lot of those trials that failed because they were uncomfortable or it didn't work or something like that, they don't publish them. So what we did was,
You don't know what doesn't work. It's like negative know-how, right?
yeah. And so we would write all these things down on that whiteboard. I remember I had a cordoned off saying, do not erase. And what I did was we started assigning to students to track down these ideas we knew had happened, but nothing ever came from them. So we did things like find people that worked at those companies on LinkedIn, and then we called them.
That's great. That's it's so brilliant, but it's so simple. The idea.
Yeah, and it was like we collected all this stuff. These are the things you cannot do.
But you know what Edison said? He said, I didn't fail. He said, there were 10,000 ways now I know that don't work. I'm that much closer to a solution.
Exactly. Well, that's exactly. I love that quote because that's exactly what we were doing, and that turned into. I can say now turned into. We're talking about what J&J has demonstrated with this technology is that 83% complete response. Like 83% of the people have no trace of cancer at the end of the therapy. I don't know what the FDA is going to do, but it's kind of hard to imagine they want to approve that because it's very well tolerated and all that kind of stuff.
That's impressive. Now, you were involved in an ovarian cancer drug distribution
In the peritoneum.
in the peritoneum. Yeah. I happen to be, unfortunately, familiar with the nature of ovarian cancer, but how did you get involved in that? What made you think do something along those lines?
I have a family member who is a ovarian cancer survivor. So it's a big unmet need. It's a very, very interesting disease in the sense that if it were caught early, it's actually the most chemo-responsive cancer. But unfortunately, it's caught too late because it's often asymptomatic until it's quite far along. It's another one of I have what I call a kind of an approach that is a theory of change. That is, I want to treat disease in individual compartments. And the reason why is, having been involved in drug development, a big part of drug development is you administer a drug systemically and hope that it acts locally. And if you design a molecule to do that, it poses quite a big constraint that
on the body.
It's going everywhere in the body. And so the idea is that why not, the disease is confined to a compartment, why not treat just the compartment? And so I get involved in a lot of that. So the bladder, the peritoneum, which is the ovarian cancer case, the brain, all of these are single compartments. I'm involved in the eye. These are individual compartments that there are procedures that you can deliver the drug to that compartment. And part of the design becomes designing a product that can go into an existing procedure that allows that procedure to do something that nobody ever intended it to do. That's how we do it. So in the urology case, we looked at the most common procedures, which are catheterization and cystoscopy. And we said we wanted to design a product that could go into those procedures in such a way that the urologist can look at it and They don't even have to read the instructions. They know inherently what to do. That is the kind of approach that we take in all of these. In the ovarian cancer case, we haven't gone beyond preclinical studies there, but we do see the possibility of being able to just treat within the peritoneum and have some pretty interesting effects, in the sense that we can knock down the cancer and completely eliminate the cancer in the peritoneum. So we've seen that in animals. Now the question is, can we develop it to a point where it's safe and effective to do in humans?
Michael, you've been MIT faculty advisor to Lemelson MIT since 2009. LMIT helps to educate the next generation of inventors, including about the critical role of IP rights. Please tell Understanding IP Matters listeners about LMIT's activities.
Originally, it was working with high schools across the country.
Okay. And then there was an Inventor Award, which I think Bob Langer may have won one year, but I think they're doing it differently today.
That's right. Well, in fact, the first thing that the Lemelson Foundation funded was an award, which Bob was one of the winners. And it was basically, I think the idea was to make inventors be like sports stars, give them the recognition. They were quite successful at that. When I joined as a faculty director, it was on the way to converting to more exposing younger people to intellectual property and how that could impact their choice of what to study, what to become, their choice in careers. And so I look at it as it's become really a vehicle for project-based learning. And the project is the hardest part of the projects is deciding what to work on. And what problem do I address? And how do you create a new product? Part of that, creating a new product, is intellectual property. The biggest part of it is I like to say to students is you have to get into the shoes of the user. You have to see the world from somebody else's perspective, and that's not easy to do. You get there by practice, and so they get practice early on. We've done thousands of students now over the a couple of decades, and I've seen it change careers. A lot of the students we work with have no interests in going into engineering or science or STEM or anything like that. And all of a sudden they realize that they have a skill that a lot of their classmates that are into science don't have, and that is understanding what people want and like what problems they have. So even just that kind of skill set, and all of a sudden we have one group of students there, like they're going to be coming to MIT. They applied to MIT. A lot of the students that we deal with don't even think about going to college. They're going to MIT because they discovered things like that mechanical engineering actually, for example, involves designing new products. And they discovered that they like that.
That's wonderful. And I've worked with Lemelson MIT and with Stephanie Couch, and it's a great team and a great program. They really do fantastic work. So it's a bit of a loaded question, but I figured I'd ask it anyway. Where did you grow up, Michael, and what were your early influences?
I grew up in Northern California. I went to Berkeley as an undergraduate and graduate student. I was a chemistry undergrad and a chemical engineering graduate student. I grew up in a place called Lodi, California.
Oh, sure.
That's where I went to high school.
There's a song.
Stuck in Lodi. Yeah.
Stuck in Lodi.
Creedence Clearwater Revival.
That's right.
Now it looks so different than what it looked like when I was there. I mean, it was much more rural. Now it's kind of suburbia. I had a lot of jobs as a kid that were working outside, and I worked on a ranch. I worked for an engineer, a civil engineer for a while. And yeah, I got used to working with my hands. My grandfather was, I guess, an inspiration in working with my hands. And so I always was building things, and I still build things. And I guess that was probably the biggest influence in going into engineering.
What advice would you have for students who may be interested in research or entrepreneurship who want to pursue technology?
Spending time thinking about problems, developing the skill sets to think through, how another person addresses problems and helping them. It's a great way to learn how to develop products because that's the hardest part of developing a product, I find, because I've got all kinds of technical solutions. I just don't know what problem to apply them to. I'd like to say, there are three main attributes to great inventors. The first is curiosity. Curious people are collecting solutions to problems they haven't encountered. I mean, that's what you're doing. And curiosity is a skill, right? You have to learn how to be curious. And I kind of look for nonlinear phenomena and collect those in the back of my brain.
What are you most curious about?
Anything that's really nonlinear, because if I can take two nonlinear things and put them together, then I've got something exponential. That's what I look for. If I take two things and they add linearly together, that's obvious, right? It's like the invention is something that you add two things together and you get an exponential. That's the non-obvious part. The second skill is kind of goes to what I was talking about with people getting into the shoes of somebody else, the skill of empathy. You have to learn how to do that. And because it's so easy to bring your own biases into a problem. You have to be aware of those biases and be able to detect those. And so that's curiosity, empathy. And the final one is actually the hardest one to teach or for especially at universities is leadership. Leadership is also a skill that you have to practice. I mean, if you think about entrepreneurs, what are they doing? They typically don't have a lot of money, right? But they have sort of an infectious enthusiasm that gets people to help them without having to be paid. And those are the three things I tell students, it's curiosity, empathy, and leadership. That's what you should get from your education. It's not a degree in chemical engineering, curiosity, empathy, and leadership.
That's terrific. And it's great that despite being so immersed in this highly technical field, you're able to pull back.
When I told that to somebody, those three things to somebody, they looked at me and said, none of those have to do with what grade you got in calculus. I said, you're absolutely right.
Well, that's why, like, great musicians or great songwriters aren't necessarily great musicians, and great inventors are not necessarily, highly educated. Sometimes they are, but sometimes they're not.
Sometimes they're not.
You are highly conversant in language and strategies of IP. How did you learn about it? Was it working with lawyers?
Yeah, I think the first few times I was sort of passive in the whole process. I mean, I basically write a technical paper and give it to a lawyer and they would come back with a draft for me to review. And I looked at it and said, okay, it looks sort of like my paper, but there's all this other stuff.
A lot of claims, yeah.
Well, then the claims. Yeah, then the claims. The first time I saw claims was I didn't really know what they were trying to achieve. And it took me a while. And I think part of it really gelled with me in the first expert witness cases that I mentioned earlier was now I understand this, like what we're trying to achieve with these claims and how the strategy is to be as broad as possible, but not so broad that it includes the prior art.
Exactly. When I learned about patents from Kenyon and Kenyon when I was doing work with them, they said it's like a mining claim. If you stake it too broadly, it's hard to defend. But if you stake it really narrow, it's easy to defend, but it doesn't include enough. We like to ask on Understanding IP Matters our guests about your first exposure to intellectual property.
It was 3D printing.
Is it a positive or negative or neutral encounter?
It was actually the patent process was quite good. My co-inventor, Ellie Sachs, had some experience with, because his earlier career was in the early version of photovoltaics. He was quite, so he had quite enough experience that in reviewing the drafts with the lawyers, he could ask the right questions and stuff like that. So I undoubtedly learned a lot from him. That kind of helped. But what got really complicated was in the licensing process because MIT took the view, rightly or wrongly, and with our support. So, I mean, they wouldn't have done it without Ellie and I saying they were going to do field of use licenses. So we had this, it was 3D printing was a process, right? So you could employ it for doing this or doing that.
Right. So there are many fields of use, which is attractive.
And exactly. And so that turned out to be the more difficult than I think either of us thought. How do you define a field? And how do you have, 12 licensees that are not stepping on one? That turned out to be, I mean, I wouldn't say it was a bad thing. If we may have done something different if we had known all the difficulty we would have. I'm not sure, but it turned out to be good, and that a lot of those licensees were very successful, and they were acquired, and all this kind of stuff. So that was many years ago. I feel it was an unexpected amount of work. I mean, it's part of, how do you monetize a patent? Part of it is licensing, and I learned a lot in that process.
Sometimes people have to be burned. I remember Jim Ferguson, who was actually a Lemelson Award winner. Jim Ferguson, he invented the LCD. He said he was a professor at Kent State, I think physics professor, and someone company, a Fortune 500 company, burned him, and he got very smart very quickly. It's unfortunate that that's how IP education often takes place. You get smart when you have to be, but there's no good preparation for it. But with organizations like Lemelson and who I work with, the Center for IP Understanding, we hope to minimize. What's on your wish list for research in healthcare? Where you see things?
I have two projects right now that I'm spending a lot of time with at MIT. The first is a therapy, which is for epilepsy. The turns out a large fraction of patients diagnosed with epilepsy have a identifiable, they call it focus or lesion in the brain that is actually the catalyst for a seizure. I think it's like 60% of people have an identifiable reason for having epilepsy. Downside is all epilepsy patients get anti-seizure medications because they really aggressively try to prevent seizures because it turns out seizures do brain damage. Neurologists say seizure begets a seizure. So you're really trying to get rid of it. Unfortunately, when you titrate patients to find the right dose, that when you get the dose of anti-seizure medication that arrests the seizures, it's often so high. Now, the reason why I mention these identifiable focus is there's actually something that can be done for those patients. They go in and surgically remove or ablate. It's probably more common now, they laser ablate it. The downside of that is, with some success, but they have to tell the patient or often the parent, because a lot of these patients are children, that there could be a neurological deficit after such a procedure. That's the first thing. And the worst part of it is they also have to tell the patient that we could take on that risk and you may still have seizures because they miss or, leave some behind or. So we identified that as the big problem. So as a result of that, only less than 14% of the patients that could benefit from this procedure actually get the procedure. So we said the reason why is because that's an ablative procedure. You can't go back if it didn't work or it caused a problem. So what we're doing is we put in a microcatheter into the lesion or drug just to that region of the brain. We've done this in animals and published it recently. and it works. Basically, we can increase the therapeutic threshold, the difference between when you benefit and the side effects of a drug, by 10,000 fold. So it's a micro amount of drug that's necessary when done. It's kind of obvious when you think about it, but we learned how to do that. So we want to do that in humans. So that's one project. The other is a diagnostic project that is, some of your listeners will probably say, well, I think there are ways to do that. It turns out there are no clinically validated ways to measure what we call hydration status, how much water is in a patient. And this is a critical number for a lot of patients. We focused on end-stage renal patients that have no kidney function. So they're getting dialysis of some type, and they accumulate fluid, and 3 times a week they're trying to remove that fluid. And you ask how much fluid to remove. Well, it turns out the nephrologist guesses, total guess. It's an educated guess, of course, but it's a guess for each patient. And they're usually trying to catch up because they realize, oh, I got it wrong. So 3 or 4 dialysis sessions later, they can change it a little bit. And so we measure that directly. It's a review right now. I'm publishing a result of a clinical trial in end-stage renal patients where we evaluate how much excess fluid they actually have.
Thank you, Michael. This has been a truly fascinating discussion. I hope our audience has enjoyed it as much as I have, and we'll have to have you back on again soon. Thank you.
Thank you.
Understanding IP Matters is brought to you by the nonprofit Center for Intellectual Property Understanding with the generous support of its partners and sponsors. Gain access to all 4 seasons by subscribing for free on your favorite platform or email us at explore.understandingip.org. Content conveyed is for informational purposes only and does not represent the views of CIPU or its affiliates. This episode was produced by Jack Shields for Wild Creek Studios. Thank you for listening.